Hello, everybody. Thank you for attending the last GreenFeed webinar in the three webinar series today. Today, we're going to talk about statistical approaches and a brief literature review of GreenFeed literature to date. Again, thank you for attending, and attendance has been quite good, and questions and our feedback has been quite good. So keep it up, and thank you for listening in. Again, this is the third part of the series. Our slides will be made available on Friday. Please email us at contact@clocky.com or my own email or Carla Rosen's email if you'd like to obtain a copy of the slides. We're also recording this webinar as we've recorded previous webinars, and our recordings will be made available to whoever would like to see them in the future. But you need to email us, and we can give you a link to the recording. So finally, today we'll talk about the statistical approaches. We've talked about theory and of the GreenFeed, and then in the last webinar, experimental design, and discuss some applications of the GreenFeed. In the future, we also, on May 13th, have our SmartFeed webinar series, where we learn more about our SmartFeed line of products and the research that's been completed using the products. Again, the SmartFeeds are a portable, practical system to measure and control feed intake. We have 4 different models that either control access to feed, feed dispensing, or on May 30th, we have models that measure ad lib intake and animal weight continuously in the field. So if you'd like to learn more about the SmartFeed products, we definitely like you to attend these webinars. Finally, I discussed last time a grant opportunity from CLOC. We're still finalizing a formal announcement, but at a high level, grant amounts will be $5,000 to $30,000, and we would like these grant opportunities to focus around using our equipment, and it's largely an equipment grant. So a strong preference will be given to institution who cost your salaries, facilities, and so forth. We also would like the topics to focus on increasing the efficiency and sustainability and productivity in production environments. So for example, how can this new high-tech equipment help producers become more efficient and actually produce a return on investment from using equipment like this in production systems? So we'll be requesting 3 to 5 page proposals with some sort of benefit to producers as an outcome and a return investment analysis. The study periods will be 1 to 3 years, starting sometime around the end of 2012. And if you have something on your calendar, the proposals will be due June 12th. And so, like I said, we'll send out a formal announcement, and as you become interested and can think of interesting ways to use SmartFeed in your research applications that would the outcomes of your research would benefit producers, we would definitely encourage you to participate in this funding opportunity. So first, we'll talk briefly a brief synopsis of GreenFeed publications. To date, we estimate there's over 250 GreenFeed publications in the world. What we found, we sold our first GreenFeed around the end of 2010, beginning of 2011, and it took about 4 years for the methodology to become adopted and tested and validated through many studies. And so the first several years were mainly validating GreenFeed and learning how we can use it in research environments. In about 2015 and 16, after validation occurred and the early adopters started publishing, there was sort of a breakthrough where we noticed very quick increase in publication rate. And in fact, in the last couple of years, there's been easily more than 50 publications a year that have come out. And it looks like through 2020, we're on track to probably surpass previous years. In terms of publication type, just at a high level, the vast majority of publications that have been, I shouldn't say majority, but the largest percentage of publications that have been out for topics is dietary studies, where breeding publications in terms of how can we select or breed animals that emit methane differently is a much smaller proportion. But part of this is actually due to the fact that there are several people using GreenFeed for genetic work. However, the genetic work is much more time intensive in some cases. In some cases, we need to have larger studies with more animals. So I think the breeding and genetics portion of this will increase significantly, especially in the coming years as we gather more data from these long-term studies with a lot of animals. And of course, there's several method comparison validation studies and some optimal use studies, along with, again, gaining traction is physiology studies. So example, how do gas emissions represent the physiology of the animal, and how can we use gas emissions to give us more information about how the animal's functioning. And of course, there's thesis and some conference abstracts. In terms of method comparisons, I'm not going to go through each one of these, and this is a summary. We have a lot of the method comparisons compiled, but it may not be all of them. So in summary, there's several method comparisons out, and since this slide deck will be available, you'll be able to review these method comparisons as you need to. Again, here's another sheet of individual trial test experiment results of GreenFeed compared to the reference method. Probably one of the most comprehensive or all-encompassing method experiment of how GreenFeed reproduces methane emissions in the field is this paper by Pekka Huttunen, and it was published in 2019. We have links to many of these references in the slide deck, so feel free to view these later. But anyways, in a meta-analysis of chamber comparisons, so emissions are measured with GreenFeed, and then the animals are put into the chamber and their emissions are measured. Huttunen published this plot of GreenFeed results in terms of grams per day, and these would be herd average results compared to respiration chamber results. So each one of these points is a trial result of GreenFeed compared to respiration chambers. Overall, we haven't found any significant slope bias. So in other words, GreenFeed doesn't seem to be systematically over or under predicting emissions compared to respiration chambers. And in this case, there's a root mean squared percentage error of about 36. So we have at a high level some decent agreement between GreenFeed and chambers. But when you do a direct comparison with two methods, there's always some confounding effects. For example, GreenFeed have access to cattle have access to the GreenFeed in the freestall, and we measure them in the freestall with the GreenFeed. We can't really put a GreenFeed in the chamber because of recirculation effects. So we're measuring the animals at a different time. They're behaving a little bit differently in the barn environment than they are in the chamber. And in many cases, the feed intake of each of the animals can be increased in the barn where they're free roaming and allowed to eat, and oftentimes it becomes restricted or reduced in the chamber, even if under both ad lib conditions we're trying to feed them the same amount of food. So we see reasonable results, but of course there's confounding effects with different feeding behaviors and measuring emissions at different times. So another interesting study that Pekka looked at in the same paper is there's several models that have been developed, and some of them are empirical models based on results from chambers. So in other words, can we use these models predict methane emissions based on various inputs such as feed intake or feed intake and nutrient quality of the feed, or methane yield-based type models. So Pekka Huttunen in this paper compared based on the parameters in the study, such as feed intake. He predicted what methane emissions should be using each model. And then he also had the result of the GreenFeed. So this would be GreenFeed versus predicted model results. What he found is that for a large number of the models, and this is again over, I think, around 70 different treatments around the world from various green feeds in different applications with different types of animals, different types of feeding regimes and so forth. But for these studies where in the 70 cases we measured emissions using GreenFeed and then predicted emissions using various models, in many cases the GreenFeed actually really accurately predicted the model results for a large number of these models. There's a few outliers, and in this case, since GreenFeed produced minimal slope bias for many of the models and there's a few outliers, we might guess that that's differences with the model rather than agreement with the model between other models. So in other words, in summary, there was an average of about 6% RMSE error of GreenFeed versus the observed model result. And the interesting thing about the study that the authors pointed out was that when methane was prediction was compared to the data used to develop the model from the chambers, the error was about double that of the error that GreenFeed predicted compared to the model. So in other words, GreenFeed predicted the model results better than the chambers predicted the model results, with a lower error and more consistency. So the authors suggested that this is because of a standardized calibration routine of the GreenFeed. And also later in the text, they suggested that sometimes chamber results have very few number of animals, and GreenFeed studies often use larger numbers of animals. When you use a larger number of animals, the statistical uncertainty with a mean compared to a large subset is smaller than a smaller group of animals. So in other words, the GreenFeed, in summary, the author suggested that the GreenFeed is quite reliable and in fact producing more consistent results than respiration chambers. There's also been a number of other studies, not just methane, for example, feed efficiency and gas emissions. So how could gas emissions be used to determine feed efficiency without putting a monitor in the pen to actually measure feed? And also, we aren't able to really practically measure feed intake on pasture. Of course, there's different methods to measure feed intake on pasture, but especially for individual animals, some of the marker methods have been quite problematic to implement. So there's many studies out correlating methane or carbon dioxide with feed intake, and also looking at the effect of methane emissions relative to feed efficiency for dairy cows or growing cows or beef steers. And actually, there's another new paper that came out in the last few days relative to gas emissions and residual feed intake. I'm not going to go into the really fine detail findings of these papers, but it's something that has been studied and published in the last several years. Of course, GreenFeed's been used for several 3NOP studies, I think around 17 journal or related articles, and has shown that 3NOP can reliably reduce enteric methane by at least 25% with a persistent effect over long time periods. And then there's some other interesting research in terms of seaweeds and algae that have been published, and there's definitely some discussions in the academic community about how practical seaweeds could be and how effective they could be, but some of the research has shown that some seaweeds can significantly reduce methane emissions. Some specific strains of algae and seaweeds can reduce methane emissions by a significant amount. So there's also other studies that have studied, that have looked at different feed additives or diet effects. And there's so many studies that we don't have time, unfortunately, to dive into all of the research. However, we do keep a list of publications up to date on our website. So if you want to go to our website and quickly find references that have produced results using GreenFeed, we try and keep the list up to date. So the website to go to for the publications, go to sealockinc.com. There's a researchers tab, and then you can go to the publications and find a comprehensive list of publications. So one of the questions, and the questions actually came up in past webinars, is how does variability in methane relate to spot sampling results? And I think sometimes there's a lot of questions in the community about, well, you're measuring for 3 to 5 minutes, 4 times per day. How well does that result actually reflect what's going on in reality? And I think these validation studies, along with Pekka's study of GreenFeed compared to model results, shows that GreenFeed in totality compares quite well. I'm going to go through an analysis of why we can effectively measure methane for such a short period of time throughout the day, but yet come up with a very consistent result over time. So I like to think of GreenFeed as sort of a way to take a picture. So we're just sampling a small amount of the environment, and we have information coming back from each specific sample. So imagine we're taking a picture of the environment, and we take a very low resolution picture. So say we're measuring an animal once or twice or 3 times with GreenFeed. Overall, it's very difficult to tell what story the picture is showing us. We know that there's, maybe some gray parts and blue parts, but we really have no idea what's going on with the animal just by sampling a few times. But we repeat the sampling and we repeat the resolution. As we sample more and more, we start to get a better idea of what the picture looks like. Here we can, as we sample, say, 10 times or so, we can start to see some resolution, have some idea what's the picture looks like in totality. We increase the resolution further and further, and finally, we sample enough, we can know what this picture looks like. And this is actually, for those of you in other countries, this is Mount Rushmore. It's located about 30 minutes from Sea Loch. Of course, it's the mountain with 4 different U.S. presidents' heads carved into it. If you ever come visit Sea Loch, we can take you there quite easily. But anyway, so as we sample more and more, we start to get some idea of what the picture looks like. And if finally, if we sample enough, we get high resolution with a very clear picture. But what happens if we continue to sample? So instead of sampling 30, 40, or 20, 30, 40 times, we sample 100 times. Actually, as we increase the samples to a significant number, we don't see hardly any difference. This would be a 188 kilobyte bit image. This is a 247 kilobyte image. There's no discernible difference from oversampling. So we need to sample enough with GreenFeed so we can get some idea of what the picture looks like. But as we sample more and more, we're not really adding information or clarifying the picture because we know what the picture looks like well enough. So, of course, methane emissions are variable. People often mention the diurnal variability, but actually methane emissions vary all the time. They vary, of course, by year, they vary seasonally, they vary by day, they actually are variable by hour, and even within the second of what you looked at in past webinars with eructation events. So variability and statistics governs how we sample. The larger the variability, the more samples we need to create the clear picture of what's actually going on with the emissions. If the variability is low, we actually need a fewer number of samples. Through this 10-year experience using GreenFeed and talking to many different scientists and students, there's actually a lot of what we call mythology circulating, where people have either heard different things or there's ideas that really aren't true, but they highly circulate through the research environment. Of course, for most ruminant scientists, everyone knows cattle don't fart methane, that most of what methane that's emitted from the animals comes out of their mouth, but it doesn't come out the rear end. But we have heard that from a few selected students. I'm not sure if they made it through their degree program, but you do hear that from time to time. We've also heard other things like cattle only emit methane while they're ruminating and chewing on their cud. They don't emit methane when they're sleeping. So when they're sleep or take a break, they stop emitting methane. And of course, we hear all the time, and we hear a whole lot less in the last few years, but especially at the start, you can't measure with variable patterns with a spot sample. And the classic mythology that we've heard is methane emissions vary by 5-fold in a day. How can you ever measure with a spot sample? So we need to really understand what's going on with a ruminant animal in order to have a better idea of how we can sample these animals using a spot measurement. So of course, with many more animal scientists on the line, and I'm an engineer, I'm going to struggle through this, but of course, animals have a rumen, and as they eat the low quality feed that most animals can't digest, but ruminant animals have evolved to digest, goes into a rumen. And the animals aren't actually digesting the feed themselves, they're feeding microorganisms that digest the feed into something that the animal can eat. And so when the feed goes into the rumen, it needs to be in the rumen for a certain amount of time for the microorganisms to break it down into something that the animal can actually get energy from. And again, this is obvious, and I'm sure everyone on this call knows that, but we need to discuss it a little bit. This would be a picture of a rumen and the actual size of the rumen packed with feed. One fact is most of the gas coming out of the rumen is actually carbon dioxide, and it's not methane. The majority gas, about 70%, is CO2, and a much smaller percentage is actually methane. So the animals are producing both of these gases. Generally, of course, it depends on emissions rate, feed type, and so forth, but at a rate about 1/2 to 2 liters per minute for the largest animals. The animals, for example, when they go to sleep, they're feeding a vat. The vat is always, the microorganisms are reacting and breaking down the material in the rumen. They don't have cattle don't have the innate ability to quickly shut off or turn on the organisms. Of course, they can control how it's digested through rumen contractions, saliva, and etc. But on short time scales, these microorganisms in the rumen are going to produce gas, and they're going to produce it all the time. When the gas is produced, it has to come out, or the animal would quickly fill with gas and bloat and die. So no matter what the animal's doing, they're emitting gas. And another important note is that especially for forage diets, when they go into the rumen, it takes time for them to break down. And in fact, there's some modeling that presentations that I've watched that the rumen residence time can be over 30 hours for forage diets. So diets that, and most forage diets that come in to the rumen take a while to actually process the rumen and go out. And in the meantime, while they're being processed, they produce gases to some degree. So we're really feeding a vat, and the vat is continuously emitting methane. So we need to understand the diurnal variation compared to methane emissions, and that really relates to feeding behavior and methane mechanics emissions in the rumen as they're digesting and being produced. So one way we can look at that is to look at some chamber studies. So this plot is showing a plot from Crompton 2011, and we need to get used to looking at data this way because there's several slides with this axis convention. So on the x-axis is time during the day. So this is 9, 11, 17, and so forth. So this is a 24-hour time frame. And on the y-axis is the methane production rate at that time. So if the animals are fed once a day, they're fed. Of course, when they're fed, the microbes start breaking down the material relatively quickly. Of course, that depends on diet quality and so forth of how quickly methane is produced, but methane will go up and is maintained, and then methane production rates will decay again until the next time the animal eats feed. So they eat feed, methane emissions go up, and that's going to be related to the feed type, how much they eat and so forth, and then they'll always decay until the next visit. And another common statistic that we look at is the minimum methane emissions compared to the maximum. So if we divide the maximum hour by the minimum hour of the day, what is that number? And so for this once per day feeding in Crompton's study and his diet, he found a maximum emission divided by a minimum emission of about 2.5. So the maximum was 2.5 higher than the lowest emission in the day. But what happens if we feed the animals twice a day instead of just once a day? So we're feeding the animals a smaller amount, but instead of one large amount at the beginning of the day, we're feeding half as much twice a day. What we find is, again, methane emissions go up, but because the amount is smaller, the amount of increase is less. And then again, emissions continue to decay again. And actually, The ratio of the maximum emission divided by the minimum emission is about 2 in this example of twice per day feeding. And finally, Crompton in his chambers fed the animals 4 times per day. Again, you see the exact pattern, but instead of feeding one mass of feed at the beginning of the day, he fed them 4 times, or a quarter of the amount 4 times per day. And again, we notice methane emissions go up and then they decline until the next visit. But we notice that the maximum ratio of the maximum hour divided by the minimum hour is quite a bit less. It's only about 1.45. So as these animals eat more frequently, the diurnal pattern decreases with the increasing feeding frequencies. And of course, this goes back to what we discussed with variability. The smaller the variability in the diurnal pattern, the less we become concerned with visit timing and the effect of spot sampling at a specific time compared to the average result. So in a case where we have a large diurnal pattern, we need to be very concerned about sampling at all times of the day in order to minimize bias. But in a smaller diurnal pattern, we can sample significantly fewer times over the day and still have a representative average. So how do we know what methane emissions diurnal patterns really are in the environment? What we found, I apologize for this being blurry, it didn't look blurry before, but anyways, Jonker in 2016 published a really interesting study where they took the animals from GreenFeed in a free stall. They fed them at varied intake rates and frequencies. So he actually tested 5 different intake rates and frequencies. I'm only showing the results of 2. And of course, you can refer to this paper if you need more information. But in the third feed intake case, he was feeding 3 times per day at a restricted intake. So the intake didn't reach the point where it was an ad lib amount. It was as much as the animals would eat all the feed and then leave. And he fed each amount for about 30 minutes to the animals 3 times per day. And then he repeated the same test in chambers. So we have the GreenFeed results in white. And again, this is versus time. So this is hour of the day, and this is methane emissions rate. So the GreenFeed individual spot samples are in white, and the chamber results are in black. And this would be a case of 3 times per day. So GreenFeed can represent and have given actually a very clear picture of the diurnal pattern from the animals just by taking the measurements and plotting them versus time between a start time and end time. On the left here is the average emissions rate for each hour for the chambers and for the GreenFeeds. And you see over the course of the day, actually really pretty good agreement between the two. The next, the last trial he completed was with ad lib feed. And again, instead of observing large peaks in emissions with slug feeding 3 times per day, we see more consistent emissions rates on average over the course of the day. And if you look at the averaged emissions of GreenFeed compared to the chamber, we see some agreement between both. I also wanted to point out that in this study, when he put them in the chamber, the animals in the chamber feed intake decreased. So that is why you see a high bias in the freestall for GreenFeed compared to a lower emission in the chamber. It was simply because at an ad lib rate, the animals didn't eat all the feed in the chamber. So we can estimate diurnal patterns using GreenFeed in the field. So since feeding behavior and how we feed the animals affects methane emissions, we need to understand how animals eat in the environment and how it's an impact on emissions. So there's an interesting study from Kilgore where it's in a meta-analysis of several different publications that have looked at the amount of day animals spend grazing in different systems. So each one of these plots is a different publication, and this is the amount of hours the animals spent grazing in each of the different environments. What we notice is that in some environments, and the author suggested that these were lower quality feeds, animals spend up to 13 hours per day eating and grazing. In other environments where animals had higher quality feed and more availability, they tended to spend less time eating and grazing. So in a case where, maybe it's a productive, high intensity, low duration grazing, we have about 7 hours per day grazing, and in other cases, we have 13 hours a day. But in summary, the animals, even in the most productive cases, are still spending 7 hours per day grazing. They're not eating 30 minutes a day that would represent these chamber studies with a slug-fed diet. They're eating consistently, and they're spending a lot of time eating. The effect of that is reducing the diurnal pattern. In concentrated systems, like for example with freestalls, many authors have suggested animals eat 6 to 12 visits 6 to 12 times per day in freestall type environments. So animals are eating frequently. They're eating smaller amounts rather than huge amounts all at once. Of course, maybe at the beginning of the day, maybe they eat quite a bit more than later periods, but there is consistent visitation and eating to some extent over the day. But also, cattle tend to eat, again, this is from Kilgore, the balance of day versus night grazing. They tend to eat universally more during the day than in the middle of the night. They tend to rest more in the middle of the night than eat. What we find in GreenFeed, actually, so this plot shows the hour of the day and the percentage of visits that occurred in a pasture situation. We find, some similar visitation structure where the animals are visiting, maybe more in the morning when they're active and not eating quite as much at night. We've since actually changed our feeding regime a little bit where it resets at midnight. And often you will see in GreenFeed data a spike when the system resets, the animals rush to it and they get some of their ration in the early morning hours. And that has helped to some extent encourage the animals to actually visit in the night as well. And then this is a feeding pattern from a concentrated dairy facility where we see visits to the GreenFeed are actually quite consistent over the course of the day. So in Jonker's study, again, when the animals are allowed an ad lib access, the diurnal pattern becomes more consistent. And when they're slug fed, we see more variability. So some of these myths that are circulated that methane varies by 5-fold of the day are actually more a result of the way animals are fed in a chamber system, slug fed, a small maintenance diet, than actually what diurnal patterns really are in production environments when your animals are eating all the time. We've also looked using GreenFeed again. GreenFeed can pretty accurately estimate diurnal patterns. Similar result where in most of the production systems it's used, we observed maximum hour of the day divided by the minimum hour of the day of about 1.5 for methane. And we've looked at a number of different systems. This would be just an example of a concentrated dairy facility. This is an example of a pasture system where the max hour divided by min hour is 1.55. And in fact, we've looked at a number of different production systems, and the diurnal pattern has tended to vary between about the max hour over min hour, about 1.2 to 1.6. That's good news for the GreenFeed because it means that we can measure with fewer spot samples over the course of the day and still have a relatively accurate result of the total average emissions for that animal. So people say, well, did their visitation somehow bias the result? And what mathematically, what is the effect of that? And we can look at that and say some what ifs. So this is a typical diurnal pattern from a freestyle system where the maximum emission divided by the minimum emission for the day is about 1.55. In many cases, the diurnal pattern is less, but in this case, this would actually be pretty representative of a case with maybe a little more variation in the diurnal pattern. And so let's say that you get a group of cattle together, and the cattle know that you want to make good measurements with GreenFeed, but they don't want you to make good measurements with GreenFeed. The cattle want to very intentionally skew your results by only visiting GreenFeed at certain times, and they know that if they visit GreenFeed at certain times, it's going to skew your answer and give you a bad result. So what can these cattle do to actually produce an error of greater than 10%? Well, in one case, the animals would have to get together and maybe coordinate, maybe have marching orders or something like that, but they'd all have to get together and decide only to visit the GreenFeed between 7 and 10 o'clock at night in order to bias your measurements more than 10% high. So, and they would have to know, well, we're not going to visit GreenFeed at any other time. And we know if we visit between this time, it's going to bias the results high. So it's a pretty significant result. Animals don't visit GreenFeed like this. They tend to spread visitation out over the course of the day with probably a fewer percentage at night, but they're not coordinating to visit the GreenFeed only between 17 and 20 hours. So this is a really unlikely scenario. And of course, if they also coordinate and they only visit between 4 and 7, they could produce a 10% bias. But this is very unlikely, and it's not the visitation structure we see to the GreenFeed. A more likely visitation structure is they don't really coordinate and they visit GreenFeed when they can get a treat every 4 to 6 hours, depending on how you set the feed settings. But at night, maybe they don't visit the GreenFeed because as often because they're resting. So if animals didn't visit for 3 hours of the day when they're sleeping, but yet we have decent visitation, we actually observe biases and errors from diurnal pattern sampling of less than 5%. So the errors under normal circumstances from spot sampling, provided we get even some distribution of visits over the course of the day, are actually really quite small, and I would say second order effects. My experience with diurnal patterns is it's a question that many people ask. It's a myth and something that's circulated through the community, but unfortunately, it's a red herring. And I don't say this because if GreenFeed actually did produce bias, we could figure out ways to model and adjust for this. I am stating this because it's really distracting sometimes scientists from getting to the root cause of why emissions rate are different than what they expected. I think, and there's a few papers that have been circulated that have suggested that GreenFeed error is all because of diurnal patterns. And instead of really investigating what the effect could be, they just state it. There's no mathematical analysis around that. And in fact, there are other factors that were causing problems, either with the GreenFeed or the animals or the other method or so forth, that were much more important to consider than diurnal patterns. So in my opinion, when you look at GreenFeed results and you analyze them, in almost every case, if you see results you don't expect, they usually aren't a result of GreenFeed, of visitation to GreenFeed. We need to look more at calibration errors, dilution, maybe a issue with the feed intake system, maybe, or the dry matter conversion, or so forth. Really, the diurnal pattern error in most cases is going to be very small. There has been a few cases in the past 10 years where, when visitation has been highly skewed, like, for example, we have the green feed on a pasture. The animals only have access to the pasture for 4 hours a day consistently, and they cannot access it at other times, so that you could end up with results that are skewed because of visitation. But under normal use of green feed in almost every production system where animals do have some access to it, we find that the error from the diurnal patterns is actually quite small. And again, this is why, with Huttonen's study looking at the model results compared to the green feed, even on a lot of different visitation strategies throughout the world where the data was used, different visit frequencies, different animals, and so forth, we actually still find very good agreement between the green feed and the model results. So another point is, one thing that GreenFeed does really well is it produces a lot of data. And often scientists ask, well, how do we analyze this data and what do we do with it? One example of the quantity of data that's produced is with only one GreenFeed unit, we produce about 1 billion data points in a year. This would be the 1 million data points is all of the different sensors measured once per second, and then that data is archived. We then break that down, and the processor breaks it down into the eructation level, and the average green feed measuring 75 feeding periods per day would actually produce about and measure about 1.4 million eructations over the course of the year. And of course, the visits, which usually this is the start that researchers use the data for further analysis, would produce about 27,000 visits. We have people who are using GreenFeed with, maybe 125 visits per day, and they have 10 different green feeds, and they end up with very large data sets. For those of you that haven't used GreenFeed before, or been able to analyze the data, what we really call sort of the raw data that researchers usually start using is called visit data. And there's an output of RFID number, farm number, the unit that it was measured with, a start time of the visit, the end time, and the emissions rates at that hour of the day for that animal for that specific visit. So you end up basically, if you were measuring for 1 year, 75 visits per day, you would end up with a spreadsheet with 27,000 data points in the system. So it's still, even the output of the green feed still produces a lot of data, and we have to figure out how to look at this data and analyze it in a way that makes sense. And so I'm going to provide some examples from papers of how other authors have used this data. So generally what we find is this visit data, feed period data, is sometimes taken for daily or weekly averages. Often people will calculate herd average CO2 or individual average methane and CO2. And this is actually for daily emissions. Like we said in past presentations, if we only sample 2 times per day, we produce high variability and high uncertainty. So generally, the individual animal emissions are taken over a time frame of about 20 visits, so a longer time period than a day. But if you definitely weekly or bi-weekly average of individual methane and CO2 is recommended and would produce useful results. The most frequent analysis in the literature is that user will specify a start and an end time of the study, and just average the visit data results within that time period. Using this approach, you can estimate average treatment results or individual methane results for animals, or we can also compute diurnal patterns and standard errors. In a few cases, users have used rolling averages where, say, if it's a 7-day rolling average, they will take an average of all the visits that occurred the last 7 days for each animal, and then output them and determine the consistency of emissions in the rolling average over time. So here's some examples. This would be an example of a seasonal trend measured with GreenFeed, taking weekly averages. So on the x-axis is time. It's a period of about 1 year, or actually, I'm sorry, of about 4 months, August through January. And they split the group into high emitting animals and low emitting animals. And then they took the averaged emissions of the by week of the high emitters and the low emitters and plotted it versus time for different feeding strategies. So on pasture feeding, we can see quite consistent differences between high and low emitting average animals week to week. And these averages are actually very, very repeatable. And you see differences in trends follow each animal. And these trends are probably related to, for example, pasture quality or the amount of available forage. And then when they brought the animals in for indoor feeding, methane emissions actually increased. And this was probably just an effect of the animals having more feed available and eating more on indoor feeding. But you can see the quite consistent trend between high and low emitting groups by taking weekly averages. They're really, really definitive. Another example for a 3 NOP trial, again, the user took weekly averages of treatment groups. So there's a control period where what this user did is they had a group of animals in a freestall, and they were able to control the amount of 3-OP to each group of animals separately. And all of the animals used the GreenFeed. So we had different animals on different treatments using the GreenFeeds at the same time. So the control group period before the trial started, all the groups produced about the same amount of methane. They implemented the different varied 3-NOP treatments or the control group where methane stays the same. And we see a very definitive and repeatable difference between different treatment amounts of 3-NOP and some consistency over time of methane. And then the last period was when the animals were taken off 3-NOP. And here we have methane and methane yield. So taking averages of animal data over a period of a week or 2 weeks can be very effective at showing us trends over time of different treatments or animal groups. This would be another example from a tie stall from Ristoff 2015 of 3-NOP. Instead of the animals having access to the GreenFeed all the time in the tie stall, of course, they're visiting at specific times. So in this study, the author took averages of the treatment group and the control group over time and plotted them. So these are each relatively intensive measurement period for the control and treatment groups. Another way to look at averages, and there's actually this routine in the post-processing software, is to define a start date and an end date, and then determine the averaged emissions in that start and end date. One thing that GreenFeed does when it runs this algorithm is it divides the hour into specific time periods. In this case, the user took 1 hour periods, and then every measurement that occurred within that hour is averaged. So here we have the average point and then the observed points for the GreenFeed. So, and then when we take the hourly averages between the start and end point, we can plot the averages in the course of hour per day. So in this case is hour per day from 0 to 24 hours. And these are the points measured between the start and end time, and these are the average points. And from that, we can estimate the diurnal pattern of the emissions. Further, you can also, if you're worried about visitation to the GreenFeed and the effect on bias, instead of having a straight arithmetic average of all the points, so say the animals are visiting more frequently when methane is higher because they're active, we can instead average the points by hour and then take an average of the hours. And that helps to some extent reduce any potential bias from visitation on diurnal patterns. But usually what we find when we do this is there's not a significant difference between a straight arithmetic average and a time of day average emission rate, because the diurnal patterns are low enough, the variation in the diurnal pattern is low enough, and the visitation is spread out enough that we have a representative average just taking an arithmetic average. Of course, we can, other authors have done this for other studies. This would be a study by Edward Caballos Garcia, where they looked at the diurnal pattern of varied dietary treatments. And you can see really there's no clear difference in the treatments over the source of the diurnal pattern, of course. But in other cases, this is another study by Amalu up in Lethbridge. They actually plotted the visitation for each treatment at visits to the GreenFeed on top of the average treatment by hour. And here you can start to see that on some dietary treatments, methane emissions are consistently lower over the course of the day than other treatments. And so if you're using GreenFeed and you go to publish a paper and say the reviewer asks you, well, what is the effect of the diurnal pattern on the GreenFeed emissions rates? Authors usually just determine the diurnal pattern for each treatment and present that inside the paper, where it can become obvious that specific treatments are either having an effect at specific times of the day, or in this case, over the entire course of the day. This would be another example just published by Gilles Renad at INRA, where two different types of heifers are studied. So here again, we have hour of the day, we have visitation, and then how emissions are different between the animals over the course of the day. So it's important that when people ask about the diurnal pattern effect and the effect of sampling with GreenFeed, GreenFeed can estimate the diurnal pattern. It can show us when the animals are visiting, but in most cases, the bias from our sampling using GreenFeed is actually quite small, and it's really a second order effect. Another, like I mentioned before, another way users will look at data is rolling averages. So this would be an example of a high emitting lactating cow compared to a low emitting lactating cow over the course of several weeks. And then this is methane in grams per day. You can see clearly that there's a very consistent trend in rolling averages of one animal consistently emitting more than another animal. And again, the variation from the study is probably higher because they had large American Holsteins mixed with Frisian animals. So So the difference between the highest emitting and low emitting animal in his case is probably less than most dairy environments. Another statistic that we often look at is between and within animal variability. It's a very simple statistic, but it's very demonstrative of the true variability that's occurring. Basically, we usually look at the coefficient of variance, which is the standard deviation divided by the mean. When we look at it for between animal variability, we'll take the average emissions from specific animals, average those emissions by animal, and then we'll compare the average standard deviation between the animals with the herd mean. And of course, the within animal variability over the same time can be the same. You would take the standard deviation of the differences in the means from either daily or weekly or so forth, and compare that with the animal mean. So this is a simple example of within and between animal coefficient of variance. So in this case, the herd average of the different animals is about 368 grams per day. Find a standard deviation between the animals. So the animal averages about 18 grams per day, and this equates to a coefficient of variance of about 5%. And then if we look at changes over the day or several weeks, the within animal coefficient of variance in this case would be about 6%. So this might be an example of how you can calculate this statistic very quickly. Of course, statistical packages like SAS can also output this data for you without running it through Excel or something like that. And SAS is almost always used to take the GreenFeed data and then perform a statistical analysis with the data. When we look at GreenFeed variability and between animal variability, we find that the variability we find between animals is more governed by animal weight. So if we have large differences in animal weight, we probably have animals that are eating at different rates. And if they're eating at different rates, they're going to most likely produce different amounts of methane. Also, if you have differences in intake, size, diet, and so forth, many of these factors affect intake, which drives differences between animals. If you have more between animal variability and intake, the size of the animals or changes in the diet or so forth, there's going to be a greater variation in emissions. Feedlot diets or high concentrate finishing diets seem to be more variable both between animals and within animals. And I think that's because we're pushing the rumen to a state where gas emissions are not very high compared to a forage diet, and the animals are sort of sitting right on the edge in terms of rumen pH and so forth, which is driving some animals maybe to produce more because their rumen state's quite a bit different than the next animal. But that's my theory. I'm sure the animal scientists online might have an opinion as well. But generally, finishing diets are much more variable than forage diets. GreenFeed, in both the method comparison results and looking at GreenFeed variability compared to other parameters, has very consistent results in between animal variability of what we would find in chamber systems. And in fact, in a few studies, authors have suggested that the between animal variability and the variability in GreenFeed is lower than chambers or SF6, and in some cases similar to SF6. So you might ask, well, is GreenFeed more variable than other methods? It really isn't. And the second question is that, and what I mean by is not variable is when we take averages over a period of time and compare that with biological parameters or method predictions, the variability is within the range of what we would see in other systems. So if people ask if GreenFeed's variable, the answer would be it's generally in line with other methods, or in some cases lower. And if you sometimes see other studies using, say, other methods such as like sniffers or lasers, and the between animal variability is reported as quite high, that's actually a result more of the measurement method than actual observed between animal variability in the environment. So in summary, there are many publications with the GreenFeed, and those of you who have had the opportunity to publish a paper or present at a conference, thank you for using GreenFeed and publishing your results. I think we're learning a huge amount from your research. If we missed your publication and didn't put it on our website, please notify us. It wasn't intentional. People generally publish papers without notifying CLOC, and that's okay. But if you want your paper listed for sure, feel free to send it to us, and we'll put a link in our publications on our website. In totality, GreenFeed compares well with other methods, and it's suggested it's more universally intercomparable than other methods because there's the same calibration expectations between the units, the same calibration procedures, and also usually people using GreenFeed are sampling larger subsets of animals for longer time periods than they might in other methods. In almost every case, diurnal patterns and spot sampling don't create large bias in GreenFeed results. And don't assume that if there's a result, it's an artifact of visit timing. Don't let that sort of cloud and distract you from finding out the real reason why methane emissions are what they are. If you sample it with enough measurements, again, 20 to 30 measurements in, as we mentioned in other slides, simply taking an arithmetic average of the measurements between a start and end time is sufficient. When you end up with large data sets, for example, 27,000 up to, say, a few 100,000, and repeated measurements from many different animals all at the same time, it's really hard to take averages in a way that skew your results. Most of the ways that you might average the data, either whether it's individual animal by day and then averaging the day, or averaging week and so forth, any of those averages with large data sets are going to produce about the same overall answer in the end. And I would say that when you get into trouble using GreenFeed is when you have small groups of animals, maybe less than 10, definitely less than 5, and you only sample some animals a few times. Those animals with only a few visits can produce highly uncertain averages. So if some animals only visit a few times and you take an average, and then your 3 other animals in the study visited 20, that one animal average is going to skew the other results maybe significantly and really not fairly weight the averaged outcome. And so if you are sampling in that case, and even if you have larger subsets of animals, if you get animals that don't use the GreenFeed much and produce useful data, it's probably better not to include those in your analysis because the averaged emission from those animals can be inaccurate. So really shoot for the 20 to 30 measurement over the course of a week or 2 weeks in order to have accurate measurements by specific animal. So the final thing is I'd like to say thank you. Again, thank you for all the research that's been completed using GreenFeed. I think that the advances that have been made in methane measurement and methane management have been really significant, and a lot of that is, almost all of it is attributed to the scientists who use GreenFeed and their brain power and creativity around designing experiments to use GreenFeed. So thank you for that. I'd also like to thank individuals at CELOC for helping support this. Carla Rosen, I think, has initiated the organization of the webinar and made sure that everyone's received invitations and registrations. Thank you, Mike, for helping put this together. And then I'd like to, of course, thank Pat Zimmerman, who's also my father. He, with his 40 years of experience in making analytical measurements, before he started GreenFeed, and his push to develop GreenFeed has really helped the scientific community with a really powerful measurement technique. When we started this 10 years ago, I don't think there's many people in the world that would invest a significant part of their retirement savings into a machine that measured cattle burps. So thank you, Pat, and thank you for your contributions to science. With that, I'll turn it over to questions.
All right, thanks, Scott. We don't have too many questions right now, just two. The first question is, what is the variability between individual GreenFeed units? For example, is there a GreenFeed unit bias?
We have many users with GreenFeed units that say 2 units that are measuring the same group of animals. And often a critical point of our review in the data is that the 2 units are on average producing the same result. If you have 2 units side by side and they're well calibrated, the herd averages, and you have any frequency in visitation, the herd averages of both units should agree within about 5%. You should also take animals that visit the green feeds more than 20 times, average the animals, and then compare the emissions by animals between unit A and unit B, and we find high correlation between one unit and the other. I should have probably included that in this plot, but you should, if you're in the condition where you're lucky enough to have at least more than one green feed measuring the same group of animals, you should be able to find relatively high agreement between the units and also between the animals when you average the animals over the course of 20 to 30 measurements. If you have higher error than that, there's a calibration problem.
Okay, thanks for that. Next question is it also advised to simply average the measurements if cows are, for example, 8 hours in pasture and 16 hours in barn?
It depends on where you measure them and where they're visiting. If you're measuring them in the barn over 16 hours and the animals are out on pasture for the remaining amount of time, the average in those emissions from the barn are going to be much more reflective of the true herd average than the other case where you're measuring them in the pasture for 8 hours and not measuring them the remainder of the time. I would say that in any case where the animals don't have at least some ability to visit the green feed at various times of the day, and they can't visit the green feed for large chunks of the day, that those are the specific cases where you have to really look at your data and make some judgments about, is this measurement technique biased in your results, and if they are, what could be the effective bias. So what I would do is I would actually plot the diurnal pattern. I would look at the emissions of the green feed just before pasture, if they were in the barn measured, and then maybe just after, and determine how similar those are. When they are on pasture, of course, they could be eating and emissions could increase somewhat. But a uncertainty analysis could be very well easily completed to determine if the errors could be significant or not.
Okay, and the final question, unless more people mail in questions, the summarized file with the raw data is updated how often?
We try and update that daily. We're always updating our calculations and so forth, and that daily update is not sometimes getting done as frequently as we would like. That will improve over time. What we also suggest is that if you're completing a study, we have a pre-experimental checklist of items to go through. Fill that out, and if there are critical times over the study where the data needs to be done, be sure and let us know, and we can make sure the data is processed.
All right, there aren't any more questions, but I did want to remind there are some people that were having some audio issues and couldn't hear. And just a reminder that we are going to post this. So if you do want to have the link for the video, just email contact@sealockinc.com. We can send you that link. One question did just come in. What I understand is that if we are doing 20 to 30 measurements per animal, which would be about 1 measurement per hour. So do you think that that 1 measurement per hour would be enough to determine the diurnal pattern, or should we have 2 to 3 measurements for each hour over the period?
So the accuracy of your hourly average is going to be governed by the number of measurements you have within that hour. So of course it is better if you have many more measurements within that hour to compute an average. If you have 1 or 2 visits within that hour, and then you put that hour in and you average it with the other hours where you have more frequent visits, potentially that hour could also skew the results. So I think that it can, I would recommend doing averages by hour long enough that we do have measurements in every hour. Alternatively, that may not always be possible. The other approach is to increase the time of day in which you're taking averages. So instead of just taking a 1 hour average, sometimes users will take a 4-hour time slot and then average those visits within the total 4-hour time slot, which gives them a better, a more representative average because they'll have more measurements within a total 4-hour time slot than a 1 hour. The disadvantage of that approach is that, of course, you lose some resolution in the diurnal pattern. So generally, when we help clients with this, we recommend timeframes of between 1 and 4 hours. Often we use a 4-hour time frame just to increase the number of visits within a time slot.
Okay, and one final question. With this video being posted with a link, are they also able to get the slide deck and the papers and articles that you link?
Well, so first is this was recorded. You can review the recording. We're going to make the slides available tomorrow in Friday, sorry, in PDF form. So that will be available. If you want them, please email me or Carla. And so we can make the slides available that way. And then what was the last part of that?
Just the slide deck, the papers and articles that you link in.
Sorry, and then in terms of the papers, there are certain rules around copyrights and distributing articles ourselves to scientists. In other words, we're not allowed to distribute an article that's required to be paid for. We can distribute abstracts, citations, and reference graphs, but generally we don't just distribute scientific papers that are covered under copyright. So in that case, we'll give you a link to the article, and then you can use your subscription service to be able to obtain that article.
Okay, then there was a clarification regarding that 8 hours on pasture and 16 hours in the barn. This person is using two different feeders, one on the pasture and one in the barn. So I'll read that question again. Is it also advised to simply average the measurements if cows are 8 hours in pasture, 16 hours in barn, if using one barn unit and one pasture unit?
If it were me, I would plot the diurnal pattern altogether of each unit, and certainly if the units are well calibrated and equivalent, you just can take the average of all the emissions from all the units data. So if you're able somehow to have two units sampling animals in different places so that you can have some equal visitation and representation over the course of the day, certainly it's fair to average those visits.
All right, that's it for questions.
Great. Thank you everybody for listening in. And if you have any questions about GreenFeed, please feel free to contact me or support@sealockinc.com. And thank you everyone for your hard work using GreenFeed and the scientific advancements that your research has led to. Thank you. Have a good day.